Good morning to one and all, and welcome to this lecture. Today we are going to see something that is associated with enzymes, because till yesterday we were dealing with enzymes, and in that enzymes itself we have gone with coenzymes and cofactors. Now this topic will be associated with the topic is associated with enzymes itself, but these enzymes are essential, or coenzymes and cofactors are essential for modulating that particular enzyme. Means, for example, if we have an enzyme and in that enzyme, the particular substrate is there, but the time required for this particular enzyme to convert the substrate will be more if the coenzymes or cofactors are not attached. So, in a clear sense, we can say that coenzymes and cofactors are modulators or stimulators by which the enzymes reacts on its substrate and generates a product. Okay. So let us understand what are the coenzymes. Now, if you analyze this particular word, then this word itself will tell you something. Let's see here, I'll just highlight this word. Up to this, it is enzymes, correct? And what is this? This is co. It means something that is associated with enzymes. Cooperation, cooperative. Okay, this is the way we actually tell the words. So operation, cooperation, co. Co means it is a subordinate that is associated with this particular enzymes. So let us understand. Sorry. Let us understand the coenzymes. Now, what are the coenzymes? Coenzymes, the substance that enhances the action of an enzyme. An enzyme is a protein and which actually functions as a catalyst to mediate and speed a chemical reaction. Coenzymes are small molecules that cannot by themselves catalyze a reaction, but they can help enzyme to do so. In technical terms, coenzymes are organic non-protein molecules that bind to the protein molecule, that is apoenzyme, to form an active enzyme, that is holoenzymes. Means ultimately the coenzymes acts as modulators in order to stimulate a reaction and in order to enhance that particular kind of reaction. So what are these coenzymes? These are non-protein molecules, organic non-protein molecules that binds to the protein and enhances its activity. Correct? So once a coenzyme binds to the molecule, binds to the enzyme, it is called as apoenzyme. And this apoenzyme now becomes into an active form, which is called as an holoenzyme. Bear in mind, these two terms are said to be very much important. Holoenzyme, it means it contains the coenzyme as well as enzyme. The apoenzyme is nothing but when the non-protein molecule binds with a particular kind of protein, it will be called as an apoenzyme. A number of water-soluble vitamins such as vitamin B1, B2 and B6 are coenzymes. So these coenzymes, maximum of the vitamins, basically the water-soluble vitamins are there, which are helpful for binding the enzymes and activating those particular kind of enzymes. Coenzymes belong to a large group called as cofactors, which include metal ions. So cofactors is a little different parts that other than coenzymes. These molecules are nothing but they are metal ions. For example, sodium, Fe, magnesium. These are metal ions which are helpful for enhancing the activity of an enzyme. Moving forward. So if you see here, this chart will let us understand. So B1 coenzyme with thymine pyrophosphates, oxidative decarboxylation reactions are being categorized, and these are called as prosthetic group. The coenzyme class is called as prosthetic group. So all of these, if you see B1, B2, B3, B2 is riboflavin, basically associated with FAD reactions, oxidation reduction reactions. B3, B6, B12, again, one of the important cobalamin. 5-deoxyadenosine cobalamin. Intracellular rearrangements. This is very much important type of coenzyme that is associated for enhancing the reactions inside the human body. Niacin, folic acid and biotin. These are again various different kinds of coenzymes that are associated, and the vitamins serve as coenzymes. Because vitamins you saw, this is the total different class of biomolecules which are different from protein molecules. Correct? These are absolutely not at all protein in their nature, but what they do, they modify that particular kind of protein in order to activate that protein in such a way that the protein will be helpful for conducting a reaction and converting that substrate into product P in a very enhanced manner, in a very useful manner. So what are cofactors? A cofactor is a non-protein chemical compound or a metallic ion that is required for enzymes activity as catalyst, a substance that increases the rate of chemical reactions. Cofactors can be considered helper molecules that assist in biochemical transformation. The rate at which these happens are characterized by the area of the study of called as enzyme kinetics. So in enzyme kinetics, these cofactors plays a very important part that every reactions, these cofactors will help to modulate or to enhance that reaction, and it helps that enzyme to convert the substrate into product P. So essential ions, loose bound, forming metal activated enzymes, tightly bound, forming metalloenzymes. Even this is an interesting field to study. Coenzymes, as we have seen, the coenzymes part again comes with cofactors, tightly bound prosthetic groups, and loosely bound core substrates. So these are all varieties of cofactors that are associated in order to enhance the reactions. Types of coenzymes, electron transfer coenzymes. Some coenzymes function by pairing electrons or negative charges to enhance a reaction. Vitamin B2, B3, C are all precursors of electron carrying coenzymes. Vitamin B2 or riboflavin is the precursor for flavin coenzyme flavin mononucleotide or FMN and flavin adenine nucleotide or FAD. Their main function is to accept and store electron within the proteins. With vitamin B3, on the other hand, is the precursor for nicotinamide coenzymes, nicotinamide adenine dinucleotide or NAD, or nicotinamide adenine dinucleotide 2-phosphates NADP, which carries electrons between different proteins. Vitamin C and ascorbic acids acts as an electron donor, changing the process of dehydroascorbic acid. This reaction is important for production of bile acid and breakdown of tyrosine. So the electron transfer mechanisms are also being known by coenzymes. Various different coenzymes, as we have seen, is vitamins basically, and these vitamins are helpful for modulating the reactions at various different reaction points. Vitamin B12, as I told you, vitamin B12 is said to be one of the most important coenzymes. Methylcobalamin or 5-deoxyadenosine cobalamin are two coenzymes from the vitamin class. Methylcobalamin is required for the productions of methionine from homocysteines, while 5'-deoxyadenosine cobalamin has the role of changing the molecular arrangement of metabolic products of some amino acids. So again, the vitamins are said to be important. Vitamin B12 has very specific functions in modulating the reaction. Coenzyme from vitamin B5. B5 or pantothenic acid is used to make coenzyme A, the termed as CoA. This has a free sulfur containing group that is attached and move carbon compounds. Reactions that uses pantothenic acid derived coenzymes include the production of fatty acids. So for the production of fatty acids, these enzymes are helpful, and who helps them? The vitamin B5 helps them. Metabolic enzymes. The most common metabolic enzymes are ATP, adenosine triphosphate. These are 4 reactions involved in ATP. Each involvement involves the movement of part of ATP molecules. An adenosine monophosphate or AMP group is transferred in the reaction that produces 5-phosphoribosyl 1-pyrophosphate, when an adenosine diphosphate or ADP group is transferred. When glucose 6-phosphate is made, 2 reactions requires the transfer of a phosphoryl group, one to facilitate the production of S-adenosylene methionine from methionine, and the other production of glutamic acid. So ultimately, metabolic are said to be one of the important part, wherein the coenzymes are being helpful for metabolic reactions, specifically in terms of ATP formation and reduction as well. Coenzyme Q10. This coenzyme Q10 is a vitamin-like substance found throughout the body, but especially in the heart, liver, kidney, and pancreas. It is eaten in small amount in meats and seafood. Coenzyme Q10 can also be made in a laboratory. Coenzyme Q10 is the most commonly used for conditions that affect the heart, such as heart failure, chest pain, high blood pressure. It also used for preventing migraine, headache, Parkinson disease, and many other conditions. The coenzyme Q10 was first identified in 1957, and Q10 refers to the chemical makeup of that particular substance. So, coenzyme Q10 is said to be important. Basically, it is associated with metabolic functioning of the heart, liver, kidney, and pancreas, and it is helpful for the reactions to take place. The functions. Coenzyme Q10 is most important vitamins, that is a substance required for proper functioning. It prevents energies, it provides energy to the cell. It has antioxidant activities. Basically, the people will suffer from heart diseases, heart failures, high blood pressure, gum diseases, Parkinson's. The level of coenzyme 10 is being lowered, and ultimately, this particular kind of infections may take place. So that's all for today. And what we have undergone through was a process that is associated with coenzymes. So in the next lecture, we'll try to study the competitive and non-competitive type of inhibitions that is associated in terms of enzymes. Okay. So good day for all.